Hello, everyone, and welcome to our third lecture in our course on drug use and abuse. And in this lecture, we'll talk about some biology. You can't really understand what drugs do, how they work, unless you understand the brain and individual brain cells that we call neurons and the cells that support them. And that's really foundational. Drugs are chemicals, as we've already seen. And they have their impact by interacting with our nervous system. We care mainly in this course about psychoactive drugs. This is a psychology course, so we care about drugs and their impact on behavior, their impact on our psychological experience. And you can't make sense of any of that unless you know exactly what drugs are doing. I say exactly. There are some drugs that we don't know exactly what they're doing. We have some data on them, but it's incomplete. That said, we know quite a bit about the brain at this point. I'm not going to say that we know everything, but all drugs, one way or another, that are psychoactive affect the functioning of the nervous system. And so in today's lecture, we're going to talk about the basic building blocks of the nervous system and how they work. In particular, we'll talk about signaling within a neuron. We'll spend some time there. So neurons use electrical signals to get information from one part of the neuron to another. And so we'll talk about how that works. And some drugs interfere with that very basic process. And that's why we need to know this stuff. So we're going to take it slow. I like your textbook, but I think it skips a little bit too much over this. It only spends a couple pages on it. We're going to spend a week. And then we'll spend another week on the larger scale of the brain, different parts of the brain, what the brain does. So we'll spend some more time at the small scale than your textbook does. And I think that's important. I think you'll understand what drugs are doing much better by our having spent a week talking about this. First, let's get to last week's discussion questions. One question, well, there are three, was about drug use and religious practice. And so we have a history in the US at least of fairly clear drug laws. It's not a long history, it's only for the last 100 years or so, but the government has a responsibility to keep the populace safe by limiting access to dangerous substances. And people can debate how strong that responsibility is. But we have organizations like the Food and Drug Administration that test drugs to make sure they're safe, other law enforcement agencies that enforce drug laws and make sure that the laws about illicit substances are being followed. And many people would not argue that safety of the public is at some level a concern of the government. And of course, part of the government's responsibility is also to, in the US, is to allow the free practice of religion. The government is not allowed to discriminate against certain religious practices. And there are limits on that freedom, just as there are limits on freedom of speech. More on that in a second. But this can bring up a conflict, and it has in the recent past. So what does one do when part of religious practice involves the use of an illicit substance? On what side does the government fall? Does it promote the free practice of that religion? Or does it prohibit the use of that substance and thereby preclude the complete practice of the religion? And so there's a conflict here. And there was a big debate when a particular Native American church group wanted to and continue using peyote, mescaline, as part of its religious practice. And this is a long-standing part of its tradition. Its tradition of using that substance is longer than the drug laws outlawing it by a wide margin. So if one wanted to, one could make the religious practice was here first. That's not a great place to start a rational argument. Which one was here first shouldn't really matter. But there's a conflict here. And so the government mulled this over for a while. And now there's a special law that allows the use of mescaline in this particular circumstance by this particular group. And of course, as I mentioned in a previous lecture, in the 60s, there was sort of a counterculture movement that claimed that its religious practice involved the use of different hallucinogens, but wanted basically to legally use hallucinogens in the context of their practices. And the government basically said no, because your practices do not constitute a religion. And part of this was almost assuredly the fact that it was a new practice. Obviously, just by being part of a counterculture movement, their desire to use hallucinogens was not popular with society at large. That's what makes it a counterculture. But hopefully, as time goes on, we can have a more principled debate as to what constitutes a religion and what doesn't. But in this case, in the case of Native American groups, there's really little debate that these constitute religious groups. They do. Again, newer religions, there's some bias against them just by virtue of being new. But it brings up an interesting question as to what exactly constitutes a religion. Also, again, part of the government's job with its laws is to keep society safe. And so the argument against this law, allowing the use of mescaline in this particular circumstance, was that it constituted a threat, not just to the practitioners of the religion, and there it was asserted commonly that people have the freedom to choose for themselves whether they practice that part of their own religion. But a larger concern for many was whether allowing drug use in this particular context will either encourage others to use this drug, or whether the drug supply itself will leak out of the group for which it is specifically allowed. And there's not much evidence for that is happening. There are other more interesting cases where, for example, a practitioner of this religion was fired for failing a drug test. And not surprisingly, the drug for which they tested positive was mescaline, which was allowed in this particular circumstance. But the employer couldn't verify that the amount of drug in the person's system was because of religious practice. And so they exerted their right to fire that person for illicit drug use. And that case was later overturned. But it's another interesting concept because unless you're there to witness the drug use, later drug tests can't confirm whether it was part of religious practice. And so this is another big question. To what degree does making specific allowances constitute a threat to the larger population? To what degree are specific allowances fair? Open question. Other question was about perception of marijuana and marijuana use. And as we've seen previously, marijuana use has been rising slowly since the mid-90s. It's not as high as it was in the late 70s, but it is a little bit on the rise. And part of this are social factors. And one social factor is the influence of parents and peers, friends, acquaintances, especially in a school setting. And so when a person witnesses other individuals using a drug, it can create the impression that that drug is popular, that drug is safe. That's not always a correct impression, but certainly in the case of parents, if the individual doesn't fear any repercussions for using marijuana, if the parents allow or encourage its use or use marijuana themselves, that's a very different environment than a family environment where it is outlawed. Peer groups are also influential. If it's one's own peer group, obviously peer pressure can come into play. But even if it's not, just the frequency of drug use, its perception that it is common can influence usage patterns. It can go the other way too. If someone has a peer that has a negative experience or even suffers an overdose, then that can influence drug use as well. And of course, recent changes in marijuana laws are a big part of this. So marijuana is increasingly decriminalized. Even recreational use is allowed now in many states. More on that when we get to our unit on marijuana. But that is probably also feeding the increase in usage. And as we've already seen also, it's not the availability of marijuana that is correlated with its use. It is the perception of riskiness and increased usage by peers and parents, relaxing of drug laws about marijuana. These all influence the perception of risk. People's personal experiences, of course, its portrayal in the media. And so all of these things influence how risky one perceives marijuana use to be, and consequently, the likelihood that someone will use it. And the third question was about the positives and negatives of drugs. Very few drugs are either entirely positive or entirely negative. And oftentimes, the positive effects are noticed before the negative ones are. And the question is why? Well, one reason is that the rewarding effects of drugs, especially drugs that are drugs that cause the user to be susceptible to dependence, the positives come before the negatives. It takes a longer period of chronic and perhaps even heavy use before the person builds up a tolerance, builds up a dependence, experiences withdrawal symptoms if the drug is not used. So things like euphoria, the physiological effects, usually the desired effects of the drug, are experienced more or less immediately or very soon after the drug is taken. It takes a long time for the person's body to get used to the presence of the drug and become even dependent on it. So the timelines are different for positive and negative effects. There's also the fact of what's called confirmation bias. When people are trying to understand something, if they go into a phenomenon or an experience with ideas about how it will turn out or ideas about how it works, they will usually notice the evidence that supports their existing ideas more than evidence that contradicts it. And so if someone goes in expecting a good experience, they're going to notice the positive effects more. If they expect a negative experience, they'll notice the negative effects more. But most people, especially recreational drug users, don't start using a drug expecting something bad. And so just psychologically, and this is not just for drug use, this is for life in general, when someone has a certain expectation, they tend to confirm it. They tend to notice the things that confirm it and ignore the things that don't. And on a larger historical scale, we've only recently been able to apply scientific methods to drug use recently. And that means that there's been a lack of scientific study. People had cultural reasons for approving or discouraging the use of alcohol or tobacco or even caffeine. But it took the scientific method. It took the ability to run longitudinal studies to figure out the long-term consequences of tobacco use. And so part of this is historical. The positives were noticed first because, again, the negatives usually happen on a longer timeline, and both in personal experience, but also you need a rigorous empirical method to find those long-term negative effects. And it's only been in recent times that the medical community can do that, can track patients over a long period of time, can run statistics, can collect lots of data and put them together and make the public aware of them. So those are all reasons that the positives are usually noticed or known about a drug, and the negatives are less known or not known until later. So good discussion last week. Keep it up. So we will have a discussion every week, and you just need to participate every week. So there will be more questions. You can pick the question that interests you most. But the idea of these questions is not to have a right or wrong answer. They're not going to be something that you can just look up in the book or look up online and find the right answer. This is not about gaining a particular piece of knowledge. It's about considering the issues. It's about weighing pros and cons. It's about crafting an argument. And so that's really the point of the discussion questions here. They're supposed to be thought-provoking. They're supposed to be controversial. There's not supposed to be an obvious answer. So we can foster some good back and forth in our discussion forum. So again, keep it up. Hopefully we'll have a good discussion this week too. Okay, before we move on to the new content for this lecture, let's go over the quizzes from last week. So performance was very good, but I'll go over some of the more commonly missed items here. So the risk and protective factors, things that make it more or less likely that one will use drugs. One is that participating in school in general, but also extracurricular activities, make it less likely that one will use marijuana. And that's different from belonging to certain peer groups. Peers can have a great influence on drug use behavior, especially certain subcultures. If a subculture sort of rejects laws in general or encourages drug use in particular, that makes it more likely the person will use marijuana. But being involved in extracurricular activities, that brings you into contact with a lot of different people. But that in and of itself doesn't promote drug use. In fact, it seems to be associated with not using drugs. And that means that extracurricular activities are a protective factor. And one thing to keep in mind here is that these factors are correlational. It is not literally the case that if you throw someone into an extracurricular activity in which they have no interest, in which they don't know anyone, it's not like that's going to magically discourage them from using drugs. There are other correlations here there are other causal factors that are possible. It may not be the extracurricular activity itself. It could be that drug usage is just lower at schools that have lots of extracurriculars. It could be that the sort of person that participates in extracurricular activities has a personality that disposes them less toward drug use. So it's not necessarily the case that these protective factors are causes. We know they're just correlated. They could be causal, but they may not be. And another factor when it comes to marijuana use, we have to remember that research shows that it's not the availability of marijuana that has a big impact on use. It's the perception of risk. If you look at years in which people think marijuana use is dangerous, marijuana use tends to be lower. If they think it's safe, marijuana use tends to be higher. And so that sort of flies in the face of a traditional notion that if you cut off the supply, that marijuana use will go down, or if you're lax about allowing the supply of drugs to come in, that will increase use. Changing the supply doesn't seem to alter the demand that much. Changing perceptions of risk does. The other quiz, there was a little confusion on what makes drugs such a powerful economic force. And there are a number of factors that make drugs such a powerful incentive when it comes to producing and distributing them. One is that having a limited supply makes something more valuable. That's not just true for drugs, that's true for anything. And in the case of drugs like opium, cocaine, even tobacco, that can only be grown in so many places. These are naturally occurring. And so that makes the field of drug use economically appealing to someone who is open to producing drugs. If they live in the right geographic area, if they have the right skill set, that makes it more likely that the drug will be a powerful economic force. If it's a drug that anyone could obtain, that can be grown anywhere, that can be produced by anyone, well, there's not much money to be made there because if anyone can do it, you're going to have a lot of competition. If the drug is hard to produce or only grows in certain places, then you've got a near monopoly on the market and you can make a lot of money, and that provides a powerful incentive. If the drug is perishable, that makes it less likely because it means your potential market is smaller. You can't get the drug all the way to a different continent if it spoils before it gets there. So if a drug is perishable, that makes it less of an economic force. If the supply is limited, that makes it more of an economic force. And obviously, if it is highly addictive, that makes it a powerful economic force because the person who buys the product will in the future feel compelled to buy more of it. It's consumable, which means it goes away, which means you always need to be supplied with more of it. And if you're addicted, you feel compelled to get more of it. So those are all factors that make drugs more powerful economic forces. Perishability makes it less powerful. And historically, our understanding of what drugs do has been limited not by religious laws or non-religious laws, not by government crackdowns on individual drugs. Those are all relatively recent phenomena. And certainly there are religious laws prohibiting certain drugs in certain religions, but over the long view of history, those are the exception rather than the norm. In fact, a lot of religions have some form of drug use involved in their practices. Historically, it was certainly true. And again, laws governing drug use are a very recent phenomenon. So that's not really what was getting in the way of understanding what drugs do. Really the lack of the scientific method or the lack of the ability to collect and analyze data. That's what made it hard to understand what drugs are doing. And when it comes to the United States in particular, there are organizations in place that evaluate drugs, that enforce drug laws. And U.S. laws on drugs are fairly strict and fairly clear. There are a lot of them. There are laws about which substances are permissible. There are laws about what the penalties are for possessing or using these substances. And so the U.S. drug laws are clear. And again, there are a lot of them. And there are, in fact, entire organizations dedicated to evaluating and enforcing drug laws. That being said, U.S. law sometimes lags a little bit behind when it comes to synthetic drugs. There are synthetic cannabinoids, drugs that imitate marijuana, or synthetic stimulants that sometimes there's not a specific law for because the chemical is brand new. It's just been synthesized. And so there is sometimes a little bit of lag. But for the vast majority of drugs, the laws exist and they are clear. For very new drugs, it may take a little while before they come into effect, before we know the effects of a drug and what the legality of the drug is. So hopefully that clears some things up for you. But in general, good job on the quizzes. Keep it up for this week. The quizzes are always required, so keep that in mind. Okay, for our unit on neurons, the building blocks of the brain, we're going to look at the brain for the next couple of weeks, and then we'll be able to understand what drugs do to the brain. So we'll start by looking at neurons. Neurons are brain cells, the basic building blocks. And more next time, we'll mention it this time, we'll look at synapses, the junctions between neurons. And then we'll look at the brain as a whole, and that will be largely next week. Neurons and synapses are very small. Obviously, the brain is much larger. But all of these things are affected by drugs. Drugs act on a number of scales. We're going to start at the small scale and then work our way up. So particularly what we're going to do is learn about the different cells that can be found in the brain. We're going to spend most of our time on neurons, but there are other cells as well, and many of them are important for understanding what makes the brain special or understanding how drugs interact with the brain. And we're going to do a little chemistry, a little biology. We're going to take it slow, so don't worry if it's been a while since you had chem or bio. And we're going to look at sort of the environment that neurons inhabit. Obviously, the larger environment is the brain, but neurons are bathed in fluid. And that fluid contains something we call ions, charged particles. And those ions are crucial for neural signaling. And we have to understand what that environment is, what the environment inside the neuron is like, and some rules that govern how ions work. And once we do that, we can start to understand how neurons transmit information. We're going to start by looking at how information is transmitted from one end of a neuron to the other, so within a cell. We'll move on later to looking at how information is transmitted between cells. We call that synaptic transmission. Okay, broad overview. The central nervous system refers to the brain and the spinal cord. And when we're talking about drugs' effects on the nervous system, the vast majority of that is going to be in the context of the brain. We'll talk a little bit about the spinal cord, or what's called the peripheral nervous system, which is the nerves in your body, from time to time. But most of our time will be spent talking about the brain when we're discussing the action of drugs in the nervous system. So the nervous system is the brain and the spinal cord. And this week, we're going to be focusing on neurons, on individual brain cells. And these cells are distinct cells. They're discrete. They're very tiny. So this diagram is not from your book, but it shows you the scale at which we're talking. Neurons can be only a millimeter in length. Some can be longer, but for the most part, they are microscopic. So we're going to start at this very small scale, understand these basic building blocks, and then go into the different parts of the brain, the different things that the brain does. So the neuron is the basic unit of information processing. It gets a signal in, it sends a signal out, sometimes, not every time. And understanding that process is really fundamental because drugs impact that process. They could stop that signal, or they could cause a signal to occur when it otherwise wouldn't, or they could change something about that signal. So the neuron is the basic building block, and so we need to understand neurons. And there are lots of them. You have approximately 86 billion of them in your brain. So lots and lots of neurons, and these neurons connect to other neurons. We're talking on the order of 1 trillion connections between all the neurons of your brain. Your brain is the most complicated system we know of. And so it's lots of information processing units talking to one another. And those units, different regions of the brain, have different jobs. And so when a drug impacts a particular kind of neuron or a particular region of the brain, whatever job that region of the brain does is going to be affected. Okay, so here this is a diagram from your book. This is a basic run-of-the-mill neuron. And we can see it has some different parts. It is not just sort of a blob like you might know the generic cell from biology class way back when. So this is a specialized cell, and it has different parts. On the far left, these little branches, we have what are called dendrites. In fact, dendrites comes from the Greek for branch. These little branching structures. And these are the parts of the neuron that stick off of the main body of the neuron, and they connect to neighbors. They're listening. This is where the input comes in. And so dendrites, and then this neuron only has a few of them, but some neurons can have lots of them, can connect to 10,000 neighbors. So this is a simplified diagram. But it gets the point across that the neuron can listen and connect to lots of neighbors at once. The cell body has the nucleus in it. The cell body has a lot of the same machinery that other cells have. So it's crucial for keeping the cell alive. And the transcription of DNA, converting DNA into RNA, getting proteins made, is an important part of how neurons work. It's an important part of how all cells work. But the cell body of a neuron is also the collecting point for information. So all the dendrites are listening. They send their signals onto the cell body, and the cell body, in effect, adds them up. It's sort of like a voting process. If the neuron is receiving enough activity from its neighbors, if it detects that other neighbors are firing, if enough neighbors fire, send signals, then this neuron will fire too. If its neighbors are quiet, then it won't send a signal. So it's sort of listening to all the votes of its neighbors. And those votes are collected in the cell body. If there are enough yes votes, so to speak, the neuron will send a signal that we call an action potential. More on that in a minute. And it sends that signal down what is called the axon. And the axon is the long part in the middle of this diagram. And so the axon is really just a conduit. It doesn't do much information processing. Its job is really to get that signal from the cell body down to what are called the axon terminals, which we see in the far right here. And the axon terminals are what connect to other neurons. We don't see those other neurons in this diagram because we're just trying to show one at a time. But the ends of those terminals would connect to the dendrites of other neurons. Could even connect to the cell body of other neurons. We're not going to make a big deal of that distinction. But the axon terminals is where information gets transferred from this neuron to the next neurons. And so there's a kind of flow of information, what your diagram here labels as the movement of electrical impulse. And so it comes into the dendrites, it's collected and added together in the cell body, it moves down the axon, and then the axon terminals will send signals of their own. We'll see later that those signals are chemicals. The axon terminals will release chemicals whenever an electrical impulse comes to the end of the terminal. And so normally they're not releasing much of this chemical. When an impulse comes down, they do. And so information flows from dendrites to cell body to axon terminal. And for our purposes, that is the only way that information flow works. So that's what a neuron looks like. Those are its parts. But the brain is composed of more than just neurons. There are what are called glial cells, sometimes called neuroglia or just glia. Neuroglia is a little bit less common in my experience. I will refer to them either as glial cells or just glia. And these are sort of the support system. You have about as many neurons as you do glia. Your book says that they're 90 times as common. That is actually incorrect. That was the thinking for quite some time. More recent evidence suggests they're not nearly as common as we previously thought. So your book has a little inaccuracy there. We're not going to make a big deal out of it. So I already said there are about 86 billion neurons in your brain. And so there are around 86 billion glia, which is a lot. Even if it's not 50 times as many or 100 times as many, it's not a overwhelmingly glia-based brain. But even 1-to-1 ratio means there's a lot of glia in your brain. Some of these are more important than others. Astrocytes are important. So astrocyte, that astro prefix means it's star-shaped. That's why it's called that. They're shaped like little stars, these little projections that come off. They connect to neurons, but they also connect to blood vessels. So they control blood flow. They can sense when their neighboring neurons are really active. When those neurons are really active, they're going to need more energy. They need more sugar. They need more oxygen. And so it's the astrocyte's job to open up the blood vessels that are nearby, open up those capillaries, get more blood flowing to those neurons that are so active. They need the blood more. And so the astrocytes will make sure that more blood flows to that area when it becomes active. They also help to form what's called the blood-brain barrier. And these diagrams are not from your book. But on the left here, you see a typical blood vessel. And chemicals can flow pretty easily through the walls of the blood vessel. So can oxygen, sugar, small molecules. But the brain is special. And one of the reasons it's special is that there are these glia, these glial cells called astrocytes that form the blood-brain barrier. The blood vessel is not the only barrier, the only wall that separates blood from the brain. The astrocytes form an extra layer. And this is why it's harder for drugs to get into the brain than it is for drugs to get into other organs of the body. Other organs don't have this extra barrier, but the brain does, because the brain needs protecting. The brain kind of has its own immune system. What are called microglia, which just means tiny glia. So they are sort of the custodians of the brain. They clean up debris, and they're also involved in the immune response. A large portion of the immune system can't make it across the blood-brain barrier. So your brain needs its own immune response. And so the microglia are a big part of that. They will sort of gobble up debris and pathogens and keep the brain healthy. We're not going to talk as much about microglia in this course because we're not as interested in immune function. Astrocytes in the blood-brain barrier are a topic we'll see again and again. There are what are called ependymal cells. And these border what are known as the ventricles of the brain. We haven't gotten to the structure of the brain yet, but your brain has what are effectively holes, and they're supposed to be there. This is not the result of trauma. Your brain has fluid-filled cavities in the middle of it. Your brain also sits not in air. If there were just air in your skull and your brain were sitting on the top part of roof of your mouth and the base of your skull, it would collapse under its own weight. And so you wouldn't have these ventricles. But thankfully, you do. And you have these ependymal cells, and their job is to produce what is called cerebrospinal fluid, or CSF. And the CSF serves a number of functions. It cushions the brain. It provides buoyancy, so the brain floats in this fluid. Again, it's not surrounded by air. It would collapse. It's not swimming in an active sense, but it is floating in this bath of cerebrospinal fluid. And it's the ependymal cells that are glia that produce the CSF. And you're always producing more and more of it. You're always recycling it, because another job of CSF is not just structural. It also provides sort of a sink for debris and chemicals. Drugs can leach into the CSF, and the CSF gets recycled. It gets shunted into your lymphatic system and gets sort of digested. And then these ependymal cells make more. So you're always making more CSF. We're not going to talk much about ependymal cells either. Oligodendrocytes, however, are very important. They produce a substance called myelin. And in the CNS, those oligodendrocytes create this waxy substance called myelin. And it wraps around the axons of neurons. Not all neurons. Not every neuron has myelin around its axon. But many in the central nervous system do, especially those that need to cross large distances. So if it's one half of the brain talking to the other, then that neuron that conveys that information is going to be quite long. And that axon will likely be wrapped in myelin to make the transmission of the signal faster. We'll see how that works in a minute. But it's not one continuous sheath of myelin. There are what are called nodes of Ranvier, as pointed out in this diagram. They're little gaps, and those are necessary for transmitting signals down the axon. The transmission wouldn't work, the signal wouldn't go anywhere if it were one continuous sheath of myelin, so there's a break every now and again. But in the brain, in the spinal cord, oligodendrocytes are what produce that myelin. And this figure down in the bottom left is an image of an actual human brain. And you'll see that the outer part of the brain is darker than the inner part. That is the difference between what we call gray matter and white matter. You may have heard that distinction before. So gray matter is on the very outside of the brain, what we call the cortex, and it doesn't really have myelin. Those neurons are talking to each other locally. They have very close by neighbors that they're communicating with. Their signals don't need to travel far. Speed doesn't matter very much, so there's no myelin there. The inner parts of the brain are transferring signals from cortex to distant parts of cortex. They're going a long way. They need that myelin. Speed matters very much when you're going a long distance. And so the inner parts of the brain, the white matter, it's white because of the presence of this fatty material called myelin. There are also Schwann cells, which are a special form of glia. Really, they're just oligodendrocytes outside the brain. We're not going to belabor that distinction. But Schwann cells produce myelin as well. They coat axons in the peripheral nervous system. We're not going to visit them again. But myelin is important. It speeds up the transmission of action potentials, of these electrical signals. Going back to neurons. So now we know the parts. We know what dendrites, the cell body, axon terminals. We know what the myelin sheath is. We know what a node of Ranvier is. These are all the structural components. And now we're going to look at function. How does a neuron work? How does information get transmitted, in other words? So we know the direction in which information flows. It goes from dendrite to cell body to axon terminal. And those electrical signals are called action potentials. And they are electrical in nature. And electricity, just even the electricity that flows through the wall into your devices, that is the movement of charge. In the case of everyday electricity, it's really the movement of electrons. It's going to be a little bit different here, but the idea is the same. You still have charged particles that are moving around, and that movement of charge forms that electrical impulse that we call an action potential. And Specifically, these charged particles are ions. More on that in a minute. So they're not electrons like you have in standard electricity, but it's still the movement of charge. And electrical charge comes in a variety of forms. Here, we're dealing with ions. So a little bit of refresher, especially if it's been a while since you had chemistry class. So a chemical element, and we're not going to go over all the chemical elements, that's not the point here. Any chemical element, whether it's hydrogen or carbon or nitrogen or oxygen, every element is defined by its number of protons. That's what makes it that element. If its number of protons changed, it would be a different element. Hydrogen has 1 proton. Simple. It's the simplest element there is. If it gains a second proton, it's not hydrogen anymore. It's now helium. And so the number of protons defines what the element is. The number of electrons can vary. So protons are in the nucleus of the atom. So are neutrons, which we're not going to worry about here. Electrons go around the nucleus. They sort of orbit. And an atom, the element that atom is, depends on the protons. But it can have a variable number of electrons. That number of electrons can change. Electrons are negatively charged. Now, often, in fact, typically, an atom will have the same number of protons as electrons. So the charges balance out. If the protons are have the same number as the electrons, you've got no net charge. If that atom loses or gains an electron, it becomes what we call an ion. It is now a charged atom. And if it moves around, that is the movement of electrical charge. This is an abbreviated periodic table. And again, the point here is not to memorize this table. We're only going to talk about a couple of elements in this class. But on the left, for example, if you look at the leftmost column, you see a +1 at the top. Those numbers are what the net charge is for an element if it is an ion. So hydrogen, which is not very important for our purposes, but sodium and potassium are important. They're on the leftmost column there. They have a +1 at the top. When these atoms become ions, it is largely due to losing an electron and thereby having a net charge of +1. If they have the same number of protons as electrons, there's no net charge. If they lose an electron, they now have 1 more proton than electron, 1 more positive charge than negative charge. So they have a net charge of +1. Other ions, like calcium in the second column, have a +2 at the top. So when calcium becomes an ion, it is likely to have a net charge of +2. It loses 2 electrons. It's twice as positively charged as a sodium ion. I'm not going to go over all of these. Chloride on nearly the far right has a net charge of -1. It has 1 more electron than proton. So what do ions do? Well, ions are governed by 2 forces. And if you understand those 2 forces, then everything about the action potential makes sense. You can, in fact, deduce what's going to happen. So this does not need to be mysterious. It does not need to seem complicated. You have some ions, and they follow 2 rules. These 2 rules, these 2 forces, sometimes they're aligned. Sometimes they both say to the ion, effectively, move in this direction. If they agree, it's easy. The ion moves that way. Sometimes the 2 forces disagree. They push in opposite directions. And there, it just comes down to which force is stronger in that situation. And the ion will move according to whichever force is stronger. If the forces are exactly balanced, the ion doesn't go anywhere. So these 2 rules determine how ions behave. And this is a diagram not from your book. But the 2 rules are diffusion. And all diffusion means is that an ion, or really any particle, whether it's charged or not, diffusion applies to everything. Diffusion means that an ion will go from where it's concentrated to where it's less concentrated. It goes from where there's a lot of it to where there's little or none of it. So it does what we call go down its concentration gradient. It wants to balance things out. So ions won't cluster together very long. They'll spread out. They'll mix themselves evenly, just like if you've ever seen a glass of water get a drop of food coloring. At first, all the food coloring is right where you dropped it. After a while, it spreads out. It becomes cloudy. And after a long time, that glass of water is now uniform in its color. That's what diffusion is. Little particles go from where they're common to where they're not until things are spread evenly. So that's diffusion. The electrostatic force deals only with charged particles, ions. And the rule here is, again, pretty simple. Like charges repel each other. Dissimilar charges, different charges, attract. If you've heard the term opposites attract, same concept here. A positive and a negative charge will move toward each other. Two positive charges, or two negative charges, though that's not a situation we'll see in this course. Two positive charges will repel each other. What this means is that a given kind of ion, say it's sodium, all the sodiums have a positive charge. So the sodiums will repel each other. They'll also tend, because of diffusion, to go from where they're common to where they're not. We'll see how that comes into play here in a moment. But the important thing to keep in mind for the electrostatic force is that like charges repel. Okay, so you have a neuron. It's got an inside and an outside. It has a membrane. And when it's not doing anything, when it's not sending an action potential, it is at what we call equilibrium. So there are different concentrations of ions both in and outside the neuron. And again, we only need to know a couple of them. But if you think about our diffusion and our electrostatic forces, both of these forces are going to encourage ions to move in one direction or another, if they can. Now, the membrane normally doesn't allow ions to cross it. So it is the dividing line between the inside of the neuron and the outside. And normally, ions can't go across. But there are special molecules called ion channels. And so normally, an ion can't cross, but if one of these ion channels is around and it's open, then the ion can reach equilibrium. So let's think about the case of sodium. Sodium, there is a lot of sodium outside the cell and almost none inside. Well, let's think about diffusion. Diffusion says things go from where they're common to where they're not. There's a lot of sodium outside, no sodium inside. So diffusion is going to push sodium into the neuron if it can, if there is a gate open, if there's an ion channel open. So the diffusion force wants sodium to go in. The outside of the cell is also normally more positive than the inside of the cell. Like charges repel each other. So sodium wants to go from a positive environment to the most negative environment it can find. It's attracted to negative charge, it's repelled by positive charge, and so sodium, again, has an incentive, so to speak, to go into the neuron. The diffusion force is pushing it in because sodium's common outside and not inside. And it's also being pushed in by the electrostatic force. There's positive charge outside and not much inside. So if allowed to, sodium will flow into the neuron until there's a balance reached, until there's as much inside as outside and the charges balance out. This is more complicated than we need to get into, but those forces will reach an equilibrium. It might be that the electrostatic force wants sodium to go in a little bit more, and the diffusion force wants it to go out a little bit more. But as long as those forces balance, you've got no net flow of sodium, and sodium's happy where it is, so to speak. So ions reach an equilibrium. So how do we know this? Well, we can measure the charge both inside and outside the neuron using what we call an electrode. And what this does, you use a couple of electrodes, you have one outside and one inside, and you can measure the difference in electrical charge between the inside and the outside of the neuron. As I've already mentioned, the inside is more negatively charged than the outside, which is one reason why sodium wants to go in. So if you put an electrode outside and inside, the difference between the two electrodes is about minus 60, minus 70 millivolts. That's just a convention, but it says that the inner environment of the neuron has more negative charge or less positive charge, those mean the same thing, than the outside. And so membrane potential is measured in millivolts, just like you have a 9-volt battery. That number indicates the difference in potential between the two poles of the battery, the two connection points. Other batteries, like AA's, have a potential of 1.5 volts. So those are in units of volts. Here we're talking about millivolts, thousandths of a volt. Very small difference in charge, but big enough in that environment that it can move ions around. So ions come in two broad classes, positive and negative, depending on their net charge. So positive ions are called cations. This looks like it should be pronounced cations. It's cations. A prime example is sodium. Sodium has a positive charge. It is a cation. And as I've already said, it is more common outside the neuron than inside. So both forces are pushing sodium in. It just doesn't usually have a way to get in because there aren't any open ion channels. But diffusion is pushing it in. There's more sodium outside than in. The electrostatic force is pushing it in. It's more positive outside than inside. So both forces push sodium inside the neuron. Potassium is more common inside than out, but it's also positively charged. It's a cation. So it will repel other potassium ions. It will also repel sodium ions. And it feels that positive charge outside the neuron. It doesn't matter whether that positive charge is from sodium or potassium. So the electrostatic force is pushing potassium in, but diffusion is pushing it out. It wants to go from where it's more common to where it's less. So those forces balance out for potassium. Potassium's pretty happy the way things are, but sodium isn't. Sodium wants to get into the neuron. Calcium I'll mention briefly. Calcium has a net charge of positive 2, which isn't terribly important for us, but the positive charge does matter. And we'll get more on calcium when we talk about synaptic transmission. It's important for the release of neurotransmitter. Negative ions are called anions. We won't talk much about them. The chloride ion is important for transmitting signals between neurons. We'll talk about that later also. I'll just mention it here so that we know the term anion means a negatively charged anion, a negatively charged ion. Ion channels are these holes in the membrane. Normally ions can't cross. Some channels are always open. There's a potassium channel. It's always open. So potassium can flow back and forth until it's where it needs to be to balance out those two forces. Some are what we call voltage gated. And sodium has voltage gated channels. Potassium also does too. And these channels are normally closed. They're only open when the membrane potential changes. When it gets to a certain level, those gates will open. And we'll see what that means for action potentials here in a second. So here's an action potential. Here's what it looks like on top. It is a change in electrical charge. So what we're seeing here, the x-axis is time, the y-axis is electrical charge. So normally the neuron is at resting potential, it's at minus 60 millivolts, but then it gets a stimulus, this little bump here. And then it produces a spike, and that is an action potential. So the spike is a rapid increase in the membrane potential. As we'll see, this is caused by sodium flowing into the neuron, and then a rapid decrease. And this is caused by potassium going out. And then the neuron gets back to its resting potential. So at first, you have a lot of sodium outside, a lot of potassium inside, and sodium doesn't have any way in. Then it gets a stimulus, and we'll see what that means in a later unit. But when the voltage changes, when the neuron gets a little bit stimulated, then those voltage-gated sodium channels start to open. And now sodium can finally come into the neuron, what it's been wanting to do this whole time. The electrostatic force is pushing it in, diffusion is pushing it in, and now it finally has a way into the neuron because that difference in charge, that little stimulus, has opened up these channels. And that causes a positive feedback loop to happen. So as sodium flows in, that membrane potential gets even more positive. that's going to open even more of these channels. And then it becomes more positive, and even more sodium channels open up, until pretty quickly, all of the sodium channels are open. Sodium is flowing in as fast as it can. It finally gets to come into the neuron. But now, at the top of this spike, now potassium isn't where it wants to be. Normally, it's balanced to the electrostatic and diffusion forces. But now that all this sodium is inside the neuron, the inside is actually now more positive than the outside. Sodium is happy because it has balanced diffusion with the electrostatic force. Electrostatic is pushing it out, diffusion is still pushing it in, it balances out. Potassium now has both forces pushing it out. It used to be happy because the electrostatic force was pushing it in, diffusion was pushing it out. But now both forces are pushing it out because of all this positive charge thanks to sodium. So now the voltage-gated potassium channels open, and now potassium rushes out of the neuron. So when sodium rushes in, membrane potential goes up, positive charge is flowing into the neuron. When potassium goes out, the membrane potential drops very quickly. Positive charge is leaving the neuron, so the membrane potential goes down. And that's just the convention by which we describe membrane potential. The more positive charge is inside the neuron, the higher on this graph the data point will be. So as positive charge comes in, membrane potential goes up. As negative charge comes in, or more likely in our case, positive charge goes out, then the membrane potential goes down. And it actually goes a little too far. But the neuron has special machines that reestablish those concentration gradients. They push sodium out again, pull potassium in again, until things are right back where they started. So that's how the action potential works. You have, at first, a balance for potassium, but sodium wants to get in. A little stimulus opens up those gates, sodium rushes in, and then potassium is no longer happy, and it rushes out. Its gates open, potassium rushes out, and then the neuron reestablishes equilibrium by pushing sodium out and pulling potassium in, and now it's right back where it started. So this process of the action potential happens over and over again down the axon. Once the cell body initiates an action potential, it happens at one point in the axon, and then all that sodium coming in, as this diagram shows, opens up the channels nearby on the next part of the axon. Sodium flows in there, and then that creates positive charge on the next part of the axon, and then sodium flows in there when the gates open. And so you have this chain reaction going down the axon. As sodium flows in, opens up the channels nearby, and then sodium flows in at that location, opens up the next channels, and so on. But it's one way. So once the channels have been open for a little while, they can't open again. And that is what's called the refractory period of the neuron. And so this keeps information flowing one way. We already saw that information flows one way and one way only in the neuron. And so that's why information flow is one direction, because the channels that have already opened are basically tired. And so they close and they can't open again. It doesn't matter what the charge is. But the channels that are ahead of the action potential, they're rested. They can open as soon as the charge is right. And that's what happens. And so this wave of sodium flows down inside the neuron and opens up channel after channel after channel until the action potential gets all the way to the end of the axon and gets to the axon terminals. Now, without myelin, this is a relatively slow process. I say slow. It's about 10 meters a second, which seems fast, but it's about as fast as a really top-notch sprinter can run. So in theory, a person could outrun an action potential in an unmyelinated neuron. A myelinated neuron, information travels much faster. Because of that presence of myelin, myelin acts like an insulator. And so it forces all the sodium to go down toward the end of the axon terminal. Sodium can't diffuse out of the channels that are nearby. So sodium comes in at one node of Ranvier. All of that charge is channeled down into the next node because myelin keeps it from coming out anywhere else. And so it keeps the flow contained. and it forces it down in one very particular direction, and it makes it much faster. So myelinated axons transmit information much quicker than unmyelinated ones. And that's why axons that are long, that are going from one part of the brain to a distant part of the brain, or going down the spinal cord, or going from the spinal cord to the outside into the extremities of the body, those are going to be myelinated because that distance makes speed very important. For tiny neurons, speed's not that important because they don't have far to send their signal. And this is called saltatory conduction, not because it has something to do with salt, but because it's from the Latin for jumping, because the charge jumps. It jumps from one node to the next. And you cannot outrun this. This is very fast. It's race car speed. So it's not the speed of light. It's not instantaneous, but it is very fast. And that's thanks to myelin. Okay, so that will cover our topics for today. So we've seen neurons, we've seen glial cells, we've seen the different jobs they have and what their components are. We've seen the different kinds of ions. We care mostly about sodium and potassium here. We've seen the forces that work on them. We've seen the diffusion force and the electrostatic force. And we've also seen the role that ion channels play. Some ion channels are always open, but the ones that we really care about here are voltage gated. They open only when the charge is right. A stimulus comes in, opens up those voltage gated sodium channels. And we saw how the action potential works. So action potentials are a sequence of changes in the charge across the membrane of the neuron. So at first, the neuron is at rest. A little bit of stimulus opens those sodium channels. Sodium flows in very quickly in that positive feedback process. Suddenly now potassium is driven out of the neuron. So we have that up and down action of the spike of the action potential. And then the neuron returns to normal. And we've seen how myelin speeds up that process as it travels axon. So that will do it for today. Be sure to take the online quiz for this unit. Go over your notes, go over this lecture, go over the reading. And when you're ready, take that online quiz. Be sure to take it by Friday. Remember, you get 2 chances. And hopefully you have done this week's reading prior to this lecture, but if you haven't, be sure to do it. So always do the reading before a lecture. Next time, we'll move from transmission within a neuron, from one end to the other, to transmission between neurons. And the junction between neurons is called a synapse. So we'll look at the structure there, and we'll look at the process of synaptic transmission. How does information get from one neuron to the next neuron? And it's done using what are called neurotransmitters, molecules. And these molecules have different jobs. They're involved in different parts of our physiological or psychological function. And this is the main way that drugs do different things. Different drugs affect different neurotransmitters. And that accounts mainly for the difference between the different effects that drugs have. Okay, so that will do it for this lecture. Be sure to take the quiz, and I will see you next time.